Yeah, so continuing on what I was talking about, all I really want to know was about more about like gene editing in relation to medicine and all the ethical concerns that come with that. So eventually I decided to look at cystic fibrosis as an illustrative example of the potential that gene editing has in medicine today, which leads me to what I'll be talking about, how CRISPR-Cas9 can be used to treat cystic fibrosis. So first, what is cystic fibrosis? So cystic fibrosis, or CF, is the most common autosomal recessive disorder in the world. So this disease is caused by a mutation in the CFTR gene, which encodes a protein channel that is necessary for critical functions of the body. And when the CFTR gene is mutated, these functions are affected, which can severely affect the digestive and respiratory tract of a person. And a patient will present CF if they inherit two copies of a mutated CFTR gene. So symptoms of CF include a multitude of conditions, and there are many fatal complications that can occur with CF. This disease can affect nearly every organ in the body, and the average lifespan of a patient diagnosed with CF is only about 40 years. So here are the 7 different classes of CFTR mutations, and each of these classes have different kinds of mutations that fall under them. So with so many kinds of CF mutations, it's very difficult to treat CF, as a person would need to inherit 2 mutated copies of the CFTR gene, which means that they could present multiple classes of CF. So modern drug therapies work to ease the symptoms of CF, but they come with many limitations. None of the drug therapies are permanent solutions, and some classes of CF mutations cannot be affected by drug therapies. So this is where gene therapy can come into play. So here's an introduction to CRISPR-Cas9. CRISPR-Cas9 was first found in the immune system of prokaryotes, preventing viral attacks by cleaving DNA. So CRISPR encodes for the RNA for Cas9, which is the enzyme that can cut DNA. So the process here in this picture is a basic representation of how CRISPR-Cas9 can cleave DNA. Once the RGN is in the correct place, it will introduce a double-stranded break in DNA, which will allow for the cleavage of DNA. So this system here has 2 RNA molecules that bind to Cas9, the CRISPR RNA or cRNA and the tracer RNA or transactivating CRISPR RNA. Viral sequences of DNA that are complementary to the cRNA are targeted if they're next to an NGG sequence of DNA, where N is any nucleotide and G is the nucleotide guanine. So how does that all help us treat CF? You can't just inject CRISPR-Cas9 into the human body and expect it to cut out mutations. You'd have to transport CRISPR-Cas9 into the affected cells. So there are many ways that you could deliver CRISPR-Cas9 into the cells. One way to do this is with plasmids, which are circular rings of DNA that are often found in bacteria. The gene for CRISPR-Cas9 can be encoded into a plasmid, which can then enter the cells and allow the cell to replicate the CRISPR-Cas9 and proceed with the editing process. So this strategy has many disadvantages though. For example, plasmids often have the gene for antibiotic resistance, which would be very harmful if introduced into the immune system of a patient. But in this case, edits would be highly specific. mRNA and ribonucleoproteins, or RNPs, can also be used to deliver CRISPR-Cas9 to the cells, and these systems have high rates of efficiency. With this method, cells can be edited inside or outside the body, which is one of its benefits. Another thing we'd have to consider is where to get cells from for the edits. Edits would need to occur in specific cells to take effect. So one of these cells is epithelial cells from the lungs. These cells will be edited outside the body and then reintroduced. The efficiency levels of this treatment are often low, as the edited cells don't always repopulate, and the cell collection process can also be very harmful to the patient due to the types of chemicals that are used. Intestinal stem cell organoids are organ-like tissues used to model diseases, which can also be used. These cells will be edited outside the body and reimplanted, and similarly, they have low efficiency rates. Induced pluripotent stem cells of a CF patient are another place to get such cells. iPSCs are able to differentiate into any cell. And in one study, these cells were implanted into patients after editing, and they differentiated into functioning lung cells. Embryonic stem cells can also be used in a similar fashion to iPSCs. So that's all really detailed work in the very cells of a person. So how would that actually treat someone with CF? This picture here demonstrates how the process would work. So first, you would sequence the CFTR gene and compare it against any known mutations. There are many CFTR mutations, and sometimes the different mutations can require different edits. Second, once the mutation has been identified, a specific repair template must be designed for that mutation. Third, cells from the patient must be isolated using some of the methods previously mentioned. And then 4, the edit must be performed using one of the approaches described earlier as well. And then it has to be validated to ensure that the edit has actually occurred. 5, once this step has occurred, the cells can be transplanted back into the patient. And then as the gene that causes CF would be corrected, the patient would potentially be cured of cystic fibrosis. So this new technology is very promising and can help save lives. However, there are many ethical issues that come with gene editing technology. For example, edits in the germline are highly controversial today. These are edits to the reproductive cells of a person or in embryos. And while this could offer a parent the chance to give their children a life without disease or disability, any changes made would be permanent. And the child has not been born yet and can't give its consent. In this case, should the parent be allowed to speak for the unborn child? Additionally, the idea of eugenics comes about. Eugenics is the purification of a gene pool, and CRISPR-Cas9 will make this possible on the fundamental genetic level of a person. In all countries abiding by World Health Organization guidelines, germline editing is forbidden. However, when technology has the potential to do this, should we still use it even to save lives? So far, the strategies to treat CF using CRISPR-Cas9 are not viable in a clinical setting. Before these treatments can be used in humans, they have to be proved safe with high efficiency and accuracy rates. They would need to be able to treat the different classes of CF mutations, given that there are so many, and they would also need to be permanent. The biggest reason that researchers have looked towards CRISPR-Cas9 as a potential treatment is the potential for a permanent solution. And while CRISPR-Cas9 cannot be used on humans yet, the possibilities are clearly endless. Very soon, it may be possible to actually cure cystic fibrosis using CRISPR-Cas9. So thank you. That's the end of my presentation.
Thank you, Nandita. Do we have any questions from the audience? Feel free to type in the Q&A or you can speak out loud. You can raise your hand and I can call on you. I have a question, which is, what was the inspiration behind getting into this topic area? I know you might have briefly touched on it, but why cystic fibrosis?
Yeah, so I wanted to learn more about gene editing in general and how that can be used in medicine because there's so many applications that gene editing could have in the modern world. So I just want to know specifically about medicine, and I wasn't really sure about which disease I wanted to learn about because there are so many that affect so many people in the world today. So I picked cystic fibrosis because it's an illustrative example of a disease that can be treated with CRISPR-Cas9. And it's a disease that has so many complications. And as I said, like the lifespan of a person with CF is only 40 years today. And so
Yeah.
I thought it was just a disease that could really benefit from CRISPR-Cas9.
Got you. And what would you say was like the hardest part of doing this research and working on this?
Yeah, the hardest part is that none of these treatments are viable today. So it was really difficult for me to get like a straight answer of like, how do we use CRISPR-Cas9 to treat this? Because there are so many methods that can be taken. You could edit epithelial cells, embryonic stem cells, the induced pluripotent stem cells. There are just so many approaches that can be taken. And like I said, none of them have been proven viable in a clinical setting. So it was just really difficult to see all that come together.
Yeah, totally. So if you had the opportunity to keep working on this, what would you focus on in the future?
Well, let me think. I would probably focus more on all these different methods because there's just so many. I might want to see how this could branch out into treating other diseases because there are so many genetically linked diseases in the world today. And I think just that connection between all this could be really interesting to see, and it would help so many people.
Totally. Well, thank you so much. I don't think we have any further questions. So thank you, Nandita.
All right, thank you.
Yeah, no worries.